Hello everyone, I'm here to share with you my thoughts regarding Dr. Anderson's challenges. First, there is no right answer for these questions because it's a matter of balancing internal and external validity determinants, as Icaro said in his post. So to start, he needs to define his target population, and if his target population are made with patients with rheumatoid arthritis condition, so he needs a criteria. And I agree with Maria del Carmen, Carolina, and Ahmed that the criteria should be the American College of Rheumatology 2010 criteria. After that, we need the inclusion criteria that would use patients who fulfill the rheumatoid arthritis criteria, and I would also add patients with moderate to severe status of disease, as assessed by another publication from the American College of Rheumatology, recommendations for disease activity measures for using clinical practice. And I would agree with Laura that in not limiting at this stage to only study patients with difficulties in achieving good results with the current treatment. The exclusion criteria, we should exclude the comorbidities which could mimic some of the symptoms, and clear inclusion criteria and definition criteria can help with this. And I would also add that you should avoid medication interactions with some class of medication, and because we already have the results from phase 2 studies, we have pharmacodynamic and pharmacokinetic data, so we can understand these possible pharmacological interactions and avoid certain types of medication. To improve our internal validity, we should avoid these possible confounding factors, and we should also avoid biases. And one of the options that we have to avoid biases is being very careful with the sampling technique used. And to improve our external validity, these sampling techniques are also very, very important too. And I agree with the colleagues that the study should be in remote centers, and it would be better and it would be easier to have more patients with the normal variations of the reference population, the real reference population, and it should be less difficult to enroll patients for the study. And I would recommend a probability sampling technique. At this point, I would like to agree with Maria del Carmen, who suggested us to use a stratified sampling technique, but I would perform slightly different. I would recommend a stratified disproportionate sampling in order to be able to quantify results according to the strata, because the disproportionate sampling technique, with this technique we can assess correctly the weight in that the outcomes have in each of the different strata. For example, we could separate the target population in two different strata, the moderate and the severe disease. Then we would randomly select samples with adequate size of moderate and severe disease patients. After that, we perform our study and we get scores as results, as outcomes. At this point, we calculate the proportional weights to each strata in the real population, and we use these mathematical values to assess the study scores, the outcome scores correctly. This is my suggestion for the sampling. I would add that after selecting the population, our intervention would be, I agree with Ahmed, disease modifying standard therapy plus the placebo, or a standard therapy plus the investigational new drug we are trying to test. So maybe could be the use of disease-modifying anti-rheumatic drugs like methotrexate, plus the investigational new drug, or another disease-modifying, or methotrexate and placebo. But I read an article who was made after a phase 3 study for an anti-rheumatic drug that compared the standard therapy with disease-modifying anti-rheumatic drugs plus placebo, disease-modifying anti-rheumatic drugs plus the investigational new drug, and disease-modifying anti-rheumatic drugs and another biological response modifier drug, which is currently used at the market. I don't know if it would be feasible for Dr. Anderson, and it's another possibility for him. Well, I would not use the placebo alone versus the investigational new drug, because since the placebo showed, the investigational new drug showed 50% of improvement in results compared to placebo, it's not ethical anymore to use this medication versus placebo alone. So my control, as I said, would be the disease-modifying anti-rheumatic drugs plus placebo, and maybe the disease-modifying anti-rheumatic drugs plus a response modifier already used for the severe disease, for example. As outcomes, I would try to define if our investigational new drug plus the disease-modifying anti-rheumatic drugs commonly used are superior to the placebo. And again, I'm not sure if I could at the same time compare to a disease-modifying anti-rheumatic drug with another biological response modifier or currently used for the severe disease. This is my question. In that time, I agree with Ahmed that the time should not be long. He said something about 2 years, 1 to 2 years in my opinion. Thank you very much.